the Canadian report, the Canadian DNA report made by Lakehead University, the Paleo DNA laboratory. This is a high-end report. This is a very good quality report, and as you can see, this is final report. This is the final report of a very good quality university test. And this is a mitochondrial DNA test, a real mitochondrial DNA test. I explained that to identify different species, we need to make a mitochondrial DNA test. Then, date is May 4, 2017, this year. The expert's name is Stephen Fratpitro, MSc. Items examined. There are 2 items examined. Were submitted by the person Terry Yamin. There are 2 samples, and the first sample is, this is the first sample of possibly biological material from a cranial brain. And the second sample is bone and tissue, is sample of bone extracted from a hand, possibly non-human. This is a final report of a very good quality laboratory from a real Canadian university. The examination requested is ancient DNA analysis, extraction of DNA, mitochondrial and nuclear DNA feasibility test, universal identification, and sex identification. Then, now we can go to the results. Then here, the results. Then I explained the 12S region and why it is used to identify different species. And I explained why this 16S region from the mitochondrial genetic material is used to identify different species. This is mitochondrial genetic material, and this is the part to test to identify if Maria is human or is not human. 12S region and 16S region. Then, first, a DNA sequence could not be produced for the universal 12S mitochondrial region from both samples. Remember, we have 2 samples. One is cranial brain, and second is bone from the hand, extracted from the mummy of Nazca with name Maria. Again, and now the 16S region. A DNA sequence was produced from the biological material from the cranial brain. The cranial brain is the first sample, remember. Then the results of the final report, remember, Lakehead University Paleo DNA Laboratory, very good quality laboratory. The closest match of readable 16S sequence was identified as a 100% match to Homo sapiens human. With this information, Maria is Homo sapiens human, and this is the final report of made by a very good quality university, the best of the best. Then a DNA sequence was produced from a bone extracted from the hand. This is the second item extracted. Then the closest match of readable 16S sequence was identified as 100% match to Homo sapiens human. Then again, Maria is human 100% match. And here we have more information. This is the mitochondrial material, and this is the 16,191 to 16,420. And here says, a DNA sequence was produced from a biological material from the cranial brain. This is the first sample. The first sample extracted from the mummy from Nazca with the name Maria. And here are the results. This sequence did contain DNA damage. The closest match of readable sequence using a genetic database was identified as a 99% match to Homo sapiens. Here, please read carefully. The 1% difference is due to DNA damage causing a mismatch. The 1% difference is due to DNA damage causing a mismatch. It means that the 1% is not related to mouse, is not related to horse, is not related to any alien. Remember, Maria was identified as 100% match to Homo sapiens, and in the results, the 1% difference is due to DNA damage causing a mismatch. Then, Maria is human. The 1% is not related to mouse. The 1% is not related to any alien. This is a final report from a very good university. And this is the best of the best mitochondrial genetic report. Then, the second sample, a DNA sequence was produced from the bone extracted from the hand. This sequence did contain DNA damage. The closest match of readable sequence was identified as 99% match to Homo sapiens human. And again, the 1% difference is due to DNA damage causing a mismatch. Then it means Maria is human, and the 1% here is only due to DNA damage. It's not related to any alien. The results: Homo sapiens human. And we have more information. Sex identification. The biological material from the cranial brain belongs to a male, not a female. Maria is female name, and that's a huge mistake. Maria is a male, not only in this Canadian report, also in the Mexican report, Maria was identified as a male. Then the name of Maria as female is a huge mistake.